This protocol enables the recording of a complete functional concentration-response relationship of GPCR agonists from one cell population and provides detailed agonist characterization even for low cell sample numbers. The main advantage of serial dosing is the increase in throughput, since a single well is sufficient for a full concentration response analysis.
This technique is well suited to study signal transduction in rare and complex biological models, like organ-on-chip formats in closed microfluidic containers. The protocol is not difficult to perform, provided the appropriate equipment is available. Preparing the various GPCR ligand solutions at the right concentration is the key to success.
Michael Skiba, a graduate student, and Anne Mildner, an undergraduate student from our laboratory, will demonstrate the procedure.
For cell seeding onto the electrode arrays, add the appropriate concentration of cells to the wells of each electrode array, and allow the cells to settle homogeneously onto the well bottoms for 10 to 15 minutes at room temperature. After at least 36 hours in a standard cell culture incubator at 37 degrees Celsius and 5% carbon dioxide, inspect the cell layers by phase contrast microscopy to ensure a complete coverage of each electrode with cells. Then replace the cell culture medium in each well with the appropriate volume of pre-warmed serum-free medium. To start an impedance recording, place the electrode array into the connecting array holder of the impedance analyzer and confirm a proper low impedance contact between the electrodes and the impedance analyzer.